Hello friends, welcome again. Today we will discuss about the neighbor slide and we will also discuss why we use a neighbor slide. So friends, we know that human blood is composed of RBCs, WBCs, platelets and blood plasma. So to count the number of WBC and RBC in our blood, we use a special type of slide which is known as neighbor slide. So friends, here you can see that a neighbor slide was invented by Kramer and it was modified by neighbor. That's why it is called a neighbor slide. And you can also see the structure of the neighbor slide, that this is the structure of neighbor slide. So friends, you can see that the neighbor slide is not a thin slide. You can see the thickness of this slide. This slide is a solid heavy glass slide. It is not a thin or double slide. It is a single solid heavy glass slide which is used to measure the number of RBCs and WBCs in human blood. So friends, you can also see the some platforms in this slide. This is the first platform, this is second platform, this is third platform. Friends, first and second platform is separated by this uplifted edge, and second and third platform is also further separated with the help of this uplifted edge. So this uplifted edge form a H shape. You can see this is a central uplifted edge, and due to this central uplifted edge, second platform is further divided into two columns. So friends, due to these uplifted edge, this whole slide was divided into three columns: first platform, second platform and third platform. And due to this central uplifted edge, the second platform is further divided into two columns, and these columns have grids. You can see this is a square like structure which is called grids. Why it is called grids? It is called grid because it have measurements which is used to measure the number of WBCs and RBCs in human blood. Friends, you can see in this diagram, if we assume that this is the cover slip, if we assume that these outlines are cover slip, then this cover slip is put above this uplifted edge. So we can say that this uplifted edge is due to is used to put the cover slip over it. So friends, we can also say that these lateral platforms are at a depth of 0.1 mm from this uplifted edge. Friends, you can see that this is the H shape uplifted edge, and this uplifted edge separate the first, second and third columns from each other, and this due to this uplifted edge, the first and third platform are at a depth of 0.1 mm, and over this uplifted edge we put cover slip. So when we put so when we measure the number of RBCs or WBC in human blood, so we firstly put the blood sample here between the cover slip and the column with the help of pipette. Then this blood sample will spread all over the column and the grid, and we used these grids to measure the number of RBCs and WBCs in human blood. So what is the structure of this grid? So friends, let's see what is the magnified view of this grid. So friends, here you can see the magnified view of the grid. You can see, if we assume that this is big square, then you can see some 9 big squares here. If we assume that this is the one big square, then it will be first, second, third, fourth, fifth, sixth, seventh, eighth, ninth big squares. So friends, these 9 big square are used to measure WBCs and RBCs in human blood. So where we count WBCs and where we count RBCs in this diagram? So friends, if I said that, if I say that, then there will be first, third, seventh and ninth, or we can say that corner big squares are used to measure WBC in blood, and the middle central big square is used to measure RBCs. So friends, these four corner squares are used to measure WBCs, and these middle one is used to measure RBCs in human blood. Friends, the length of these all these three big squares are 3 mm. If we start from here, then till here, this length of these squares will be 3 mm, and the breadth also will be 3 mm. So the area of all these 9 big squares will be 9 square. So friends, if we say that these 3 big squares will be of 3 then one big square will be of 1 Friends, you can see the structure difference in all these diagrams, in all these squares. So friends, here you can see that this one big square is further divided into 16 small squares. If we assume that this big square have three horizontal and three parallel line, then this big square will divided into 16 small squares. And here you can see that this middle big square will be divided into 25 small squares. So this is the difference between the central square from the corner squares. So you can see 16 small squares in the four corner squares and 25 small squares in the middle square. This one is used to count the number of RBCs, and these four are used to measure the number of WBCs in human blood. So here 16 small squares and here 25 small squares are used to measure used to measure RBCs in this diagram. So friends, if we say that if we say that this will be the length of one big square, then it will be 1 Here you can see that the breadth of 1 square, so length also will be 1 square of one big square. So if this whole will be 1 square, then one division of it will be 1 by 4. Here you can see that this one square will be 1 by 4 or 0.25 in length. So friends, this will be the length of 0.25 and this will be breadth, which will also be 1 by 4 or 0.25 and we know that depth will be 0.1 so the volume of this small square will be 1 by 4 into 1 by 4 into 1 by 10. So it will be 1 by 160 cube. So friends, volume of this will be 1 by 160 cube, and the square and the area of this will be 1 by 4 into 1 by 4, and this will be the length 1 by 4 and this will be the breadth 1 by 4 So friends, one square will have 1 by 4 length and 1 by 4 breadth and 0.1 depth, or volume 1 by 116 square. But here in this middle central square, if we further magnify it, then here you can see that this whole is a one central square which further have 25 small squares, and one small square further have 16 small sub squares. So the area of one small square will be 1 by 5 into 1 by 5 is equal to 1 by 25 square. So area of this whole one small square will be 1 by it will if we assume that this is 1 by 5, then it will be also 1 by 5, then area will be 1 by 5 into 1 by 5 is equal to 1 by 25 square. So friends, if this is a further small subdivision and these are 16 small squares inside this small square, so here are further 16 subdivisions in this small square. So the length of this will be 0.05 because the breadth of this will be 1 by 4 and the length will be of this whole square will be 1 by 5 and 1 by 4 will be 1 by 4 will be the breadth of this whole square. Then the length of this one small square will be 0.05 And one important thing about this is that all the four corner squares, here you can see these dark lines, this square, this whole small square, this one small square, and this small square, and this small square, and in last this small square, these five squares in the middle central square is used to measure the RBCs of blood. So these five sites at the of the corner and one of the middle is used to measure the RBCs, is used to count the number of RBCs in human blood. So friends, you can see that this is a length of one small squares 1 by 5 is equal to 0.2 mm, and this is also the breadth of this small sub squares. Then it will be 1 by 5 is equal to then it will be 1 by 5. So the area of this whole small square will be 0.04 mm square. Friends, if we assume that this will be 0.2 mm, then this will be 0.2 mm. So the area of this whole small square will be 0.04 mm square. So friends, this whole middle big square is used to measure RBCs, and these corner four squares are used to measure RBCs. And what rule is followed to measure the RBCs? So friend, here you can see this is these dark lines. So we count the number of WBCs only at the top and at the right side. At the bottom or at the left, the if the WBCC is present, then we not count them. Then we only count the WBCs which is present at the top or which is present at the right side and at the middle. At the left and bottom side, the WBCs, which WBCs are present is not counted. This is a rule for counting of WBCs, and same rule is follow for the RBCs. So friend, this was all about the measure, how to measure or how to count the number of WBCs and RBCs. These four corner diagrams, these four corner squares are used to measure or count the number of WBCs, and the central one is used to measure the number of RBCs in human blood. So friends, I hope you like this video. Thank you friends.